Welcome, everybody. Thank you so much for joining us for the first event in our special 5-part series called Live Longer, Live Stronger. This is in partnership with the Sanford Burnham Previs Medical Discovery Institute. They've been a wonderful partner for the fleet for a number of years now, and we're excited that they wanted to partner with us on this special series to help us understand what research is going on in San Diego to help us age more healthy and hopefully give us a less complicated life as we age and a healthier life as we age. And there's lots of work being done at the Sanford Burnham Previs Medical Discovery Institute. That's why it's a 5-part series, because there's lots we can talk about, and we're excited for the series. The series will take place on the first Wednesday of every month online. Just because, as you know, we cannot unfortunately go back into the building or into the community at this point as we continue to battle the pandemic, but we're glad that we can still engage with all of you online. So the event will take place on the first Wednesday of each month from 2 to 3 p.m. And at the end of the talk, I'm going to go through the list of the other topics that will be coming up in this series over the next 5 months. So with that, I don't want to take too much time away from the actual presentation, and I'm super excited to introduce to you Dr. Peter Adams.
Hi, am I on?
Yes, you are.
Okay, let me just share my screen. Okay, and great. Can you see my slides okay?
Sure can.
Okay, good. So yeah, thanks very much for joining and listening in, everybody. I appreciate it, and especially given some of other events ongoing right now, which I'm sure are competing for everybody's attention. So let's just hope everything works out okay. But what I'm going to talk about today is healthy aging to prevent cancer. Okay, so I'm going to talk to you a little bit about the relationship between aging and cancer, and then how we hope that by understanding this relationship between aging and cancer, we can perhaps intervene to prevent or delay cancer. So, in terms of the relationship between aging and cancer, I think everybody knows or is aware of the fact that the incidence of cancer increases quite strikingly with age. Okay, as shown on this slide here. And really, all you need to take away from this slide is that most of the common adult human cancers, colon cancer, lung cancer, breast cancer, etc. I mean, these increase dramatically with age, and you can see here, roughly from childhood to about 80 years old, there's approximately a 100-fold increase in the incidence of many of these cancers. Okay, so really dramatic increase in incidence of many cancers with age, and it's obviously very important to understand why that is. And hopefully I'll also convince you that actually, understanding why that is actually also a very, fundamental and interesting biological problem. Okay. So I'm going to start off by telling you a little bit about what is aging and what causes aging, and then also, what is cancer and what causes cancer. Okay, because I think we need to understand the two of them separately before we can understand how they work together. So, we're all very familiar with aging. We kind of take it for granted. We almost don't think about it. We just think it's, of course, absolutely inevitable. But what is it really? How do we define it? Okay, so aging is generally defined as the increased risk of death and disease and declining function with age. Okay, so I think everybody has noticed that as we get older, our functions become impaired. We become weaker. We're more frail. We're more prone to fractures. We're more prone to chronic diseases such as Alzheimer's, diabetes, heart diseases, and of course cancer. okay, and ultimately death. Okay, so aging is really the increased risk of all of those processes over time. What causes aging? This is a slide that I borrowed from a colleague of mine, Malena Hansen, here at SBP. And it really just summarizes how we think, or I think we know, that aging results from the accumulation of damage. Okay, and it's damage at many different levels in molecules, cells, and tissues and organs. Okay, here it's depicted within a cell, and you can see that this healthy cell from a young person is well ordered and well structured. But as we age, damage accumulates, and that leads to this obvious dysfunction and impairments of organization within cells. And so this is one type of damage which accumulates with age and contributes to the aging process and the functional decline and disease. And I'll talk a little bit more about some of these types of damage in a few slides. But the bottom line is we think aging results from the accumulation of damage. What is cancer? Well, cancer, I'm sure you all know, is a disease which is probably most fundamentally characterized as being, the abnormal and excessive proliferation of cells. And so, in a normal, young, healthy tissue, you can see that the cells in our tissues, this could be, pretty much any tissue within the body, the cells are well organized and lined up and doing their jobs properly. But for some reason, these cells can become abnormal. This tends to happen as we get older, and that can lead these cells to multiply and proliferate more than they should do, such as you get a mass like this. That mass gets bigger and invades adjacent tissues and eventually can spread to other tissues within the body and really wreak havoc throughout the body and ultimately kill us. Okay, so that is cancer. What is the cause of cancer? Well, interestingly, like aging, cancer essentially comes about through accumulation of damage. And in particular, with cancer, there's been a lot of focus upon DNA damage. Okay, so I'm sure you all know that DNA is the molecule inside of our cells, which codes for all of the functions of those cells. Well, this DNA can be damaged by various different chemicals and even just normal physiological processes going on inside the cells have a tendency over time to damage the DNA. Things like radiation and smoking and pollution can cause excessive damage to the DNA, and that's why these tend to be linked to increased incidence of cancer and other diseases. Okay, but fundamentally, cancer is thought to be in large part a disease that comes about through damage to DNA, and that damage in cancer cells is often referred to as mutations. Okay, so these are changes within the DNA which drive this abnormal proliferation of the cancer cells. But as I said earlier, okay, as well as this damage to DNA, there are many other types of damage which accumulate within cells, and I'm not going to go through these in detail, okay, but as you know, as well as DNA being damaged, there can be changes to chromosomes, proteins in the cells become misfolded, there can be metabolic changes, okay, so there are actually many different types of damage within cells, and I think probably many of these types of damage are contributing to the increased incidence of cancer with age. So really then, to summarize what I've said so far, we think that the passage of time essentially leads to accumulation of molecular, cellular, and tissue damage to molecules such as DNA, and damage to cells themselves. And this damage is what drives the process of aging, okay, which is, as I say, the functional decline which characterizes aging, but also these diseases of aging, cancer and many other diseases of aging. So in actual fact, we tend to think of aging and cancer and these other diseases as almost being essentially one and the same process. Okay, these diseases which characterize the aging process, they're all driven by the accumulation of this damage which drives aging and the diseases associated with aging. So that's why we think there's such a tight relationship between the process of aging and cancer, because essentially they are one and the same process. So that then raises the question, okay, can we slow down the accumulation of damage with age in such a way that will prevent or delay the onset of cancer? And before I try and convince you that we think this is possible, I just want to say a little bit about what we would really like to try and achieve here. Because sometimes when we talk about biology of aging and aging research, people think that, well, what we're trying to do is just make people live forever. Okay, and that's probably not a desirable thing and would introduce many problems in its own right. Okay, so in my opinion, the goal of what we're trying to achieve here is not to try and make people live forever, but actually just to get people to live more healthily. And so what do I mean by that? So here's a depiction of a lady who lives to about 80 years old, and like many of us, the last 10 to 20 years of her life might be characterized by one or more chronic diseases. So now in principle, we could make that lady live to 100 years or more, but she might still get chronic diseases from the age of 60 onwards, okay, in which case all we've done is increase the period of chronic disease. That's not a particularly desirable thing to do. So what we're trying to do in the aging research field is, perhaps extend lifespan modestly, okay, 100 years, 120 years maybe. But perhaps more importantly, is just to decrease this period of chronic disease at the end of the lifespan. So you can see that this lady now has a relatively short period of chronic disease and a much longer period of healthy, productive, happy, middle-aged life. Okay, so when we talk about understanding aging so that we can perhaps extend lifespan and delay and prevent diseases, in my opinion, the real goal should be what's depicted down here. Okay, a modest extension of lifespan, but more importantly, a much more dramatic extension of what we call health span. Okay, in other words, we would like to delay the incidence of cancer so people don't have to tackle it for 10 or 20 years. So what's the evidence that this is achievable? Well, I'm a scientist, obviously. So, as scientists, we tend to kind of go back to, the fundamentals of biology. Okay, that can teach us a lot. And if we look at biology, one thing it tells us is that, if we just look at evolution and this, so if we look at many different species and we look at their lifespans and their health spans, we can see that in actual fact, it does vary dramatically through evolution. Okay, so if you take a mouse, for example, which might live 3 to 4 years, 4 years maximum, more like 2 to 3 years, I think. A mouse can live 2 to 3 years and will typically, at least in the lab, a mouse will die of cancer. In contrast, humans, we can live up to 100 years, but we typically don't get cancer until 60 or 70 years old. Okay, so there is a dramatic difference in the age at which different species get these chronic diseases, such as cancer. And from a biologist perspective, we know that although all these species might in many ways be very different, biologically, we're actually all fundamentally very similar. Okay, we have very similar DNA, very similar cells, many of the tissues, the brain, the heart, the liver, etc. Many of them are very similar. Okay, so despite these similarities, evolution tells us that these diseases are not inevitable. We don't have to get them at a certain age. Okay, that the age of onset can be manipulated. So that's one thing that evolution tells us. There's also, and I just wanted to make this comment on this one specific example. Okay, so I've already mentioned the mouse. Okay, this is a mouse here, obviously lives typically about 3 years, and as I say, in the lab will typically die of cancer. This here is, you may recognize, this is a naked mole rat. Okay, and the remarkable thing about this is a rodent similar size to a mouse, lives underground in hot parts of the world. The naked mole rat typically lives 25 or 30 years and very rarely, if ever, gets cancer. So why is it that these two similar sized rodents, in many ways very similar, why do they have such a dramatic difference in their age of onset of cancer? Again, I think this tells us that this is a process that can be controlled if we can only understand it. And we are making progress towards understanding this, because we can make DNA mutations in a mouse, for example. This is what's known as the Ames dwarf mouse. It has a mutation within a gene called the PROP1 gene, which impairs development of the pituitary gland. So as a result of that, this mouse has low levels of growth hormone and is therefore small and I think pretty cute. But interestingly, it also lives a lot longer than its normal counterpart. So you can see that the normal mouse, about 50% of them will die after about 22 months, but for the dwarf mouse, 50% of them don't die until about 35 months. Okay, so the dwarf mouse has a much longer lifespan, but also about a 50% decrease, a much longer lifespan, but also a dramatically delayed incidence of cancer. Okay, so just by mutating one gene in the mouse, we can delay the onset of cancer. There are other experimental interventions that we can make. So this is a gray mouse lemur. This is a primate, okay, so not too distantly related from us. And like many species, okay, if we put this gray lemur onto a calorie restricted diet, okay, which means its diet is limited by about 20 to 40% of what it might eat otherwise, then you can see what happens is that the lifespan of this lemur is quite dramatically extended. Okay, so normally these lemurs in captivity will live to about 6 or 7 years, but if we limit its food intake, then it can live to nearly 10 years. And as well as extending lifespan, this dietary restriction will also delay the onset of diseases such as cancer. So I'm not going to go into too much detail as to why this is, other than to say, in very general terms, it's thought that probably how dietary restriction works is by limiting the resources available to the lemur, you essentially make that lemur invest more of its effort and its resources in kind of preservation of its tissues. Okay, in other words, damage limitation and damage removal. Okay, so it's somewhat paradoxical, I know, but paradoxically, if you limit the diet, then that encourages the removal of damage and therefore delays the onset of many diseases and extends lifespan. So that's dietary restriction. We also know that exercise in humans is good at extending lifespan and health span and delaying specific diseases such as cancer. Many different types of exercise can do this. And kind of on the flip side to what I've told you about dietary restriction and exercise, obviously the flip side of that is, too much calorie intake and too little exercise. And obviously that tends to result in obesity. And we also know that obesity is strongly linked to an increased incidence of many different types of cancer. Okay, so we can see this relationship from both sides. Okay, dietary restriction and exercise is beneficial, but lack of exercise and too much food causes obesity, and that's detrimental and can cause cancer. So, what this tells us is all these things that I've told you really say that, the incidence of cancer can be modulated. Okay, it's not inevitable that we're going to get cancer at 60 or 70 years old. It can be controlled. And so, one of the goals of, I think, healthy aging research, and particularly, if we're focused upon cancer as a disease, is to, to identify additional interventions which might work in the same way as dietary restriction or exercise to prevent the incidence of cancer. And so there are already drugs which are being considered for this. So one of them, which you're probably familiar with, is metformin. Metformin is first and foremost used to treat diabetes. But what has been noticed is that diabetics who are taking metformin have a lower incidence of various different chronic diseases, including cancer. So diabetics on metformin have a lower incidence of cancer compared to diabetics who are not on metformin. Okay, so it's thought that as well as tackling diabetes, metformin is good for preventing the onset of various other diseases of aging, including cancer. And so this is Nir Barzilai, who's been a big proponent and the leader of what's known as the TAME study, okay, or Taming Aging with Metformin. And so Nir is leading a study where normal, healthy humans will be given metformin or a placebo. And the question will be, does taking metformin in later life actually, does it prevent the onset of these other diseases, including cancer? And metformin is not the only drug that's being considered for this. Other drugs that are also discussed, which you may have heard of, are things like rapamycin, alpha-ketoglutarate, and nicotinamide riboside. So what we would like to do is identify drugs which can prevent the onset of cancer, and I think already there's good evidence that these types of drug can exist. So I'm going to talk a little bit more now about what we're specifically doing in my lab to tackle this problem. And I mentioned earlier that there are many different types of damage that accumulate with age. In particular, I talked about DNA damage, which we know is important for cancer. Another type of damage which we think is important in cancer is what's known as epigenetic damage or epigenetic alterations. And so first of all, though, I need to tell you about the epigenome. So what is epigenetics? What is the epigenome? Well, you all, of course, know that in our bodies we have many different tissues, the heart and the brain, for example. And these tissues have very different functions, and that's because the cells that comprise them are very different themselves. Okay, so a cardiomyocyte, which controls the beating of the heart, is very different from a neuron in the brain, which is responsible for our thinking and our consciousness and all those other amazing things. But this is really quite paradoxical, because it turns out that even though these cells have very different functions, the DNA inside them is actually, well, essentially identical. Okay, so this is a molecule of DNA. These are the letters, the A, G, T, and C, which make up the code in the DNA. And this sequence is essentially identical between a heart cell and a brain cell. So how is a heart cell different from a brain cell? Well, that's where epigenetics comes in, okay? Because epigenetics is what's responsible for that difference. And so specifically, what do I mean by that? Well, if you imagine inside each of our cells, there are about 2 meters of DNA, and each of those 2 meters of DNA has about 6 billion of these letters, the G, A, T, and C. So that's, you know, if you consider the trillions of cells which make up your body, okay, that's where these amazing statistics of, I'm not quite sure exactly what the number is, but it's something like if you took all the DNA from all the cells in your body, then it would go to the moon and back several times or something like that. Okay, but it's a phenomenal amount of DNA is inside your body, and as I say, 2 meters in each cell. That DNA is folded up inside the nucleus of the cell, which is that middle compartment of the cell, and it's folded up in very intricate and remarkable ways. And perhaps the most fundamental unit of folding is what we call the nucleosome. Okay, so this little bead structure here, and if we zoom in on that, then what you have is inside we have these protein molecules, and wrapped around it is the DNA. Okay, this is a nucleosome, and where the epigenetics comes in is that this DNA and the proteins around which it's wrapped they've all got various different chemical modifications on them. So the function of the DNA is not purely a consequence of the DNA sequence itself, but it's also a consequence of all these other modifications to the DNA. And so this is what we call the epigenome. These additional modifications to DNA which affect its function. And it's these modifications which mean that which are responsible from a heart cell being different from a brain cell, from a liver cell, etc. So that's the epigenome. The other thing that's, I think, really interesting about the epigenome, though, is that it's not a fixed structure. So if you think of a DNA sequence, okay, those letters, the G, A, T, and C, those we think of as being, pretty fixed, okay? Unless something goes wrong, they don't generally change. But the same is not true for the epigenome. And in actual fact, the epigenome is, we think of as being very plastic and able to kind of move around, okay? And I can show you that here. And this is an experiment done by 3 people in my lab, Kaelin, Zong, and Hiroshi. And what they have here these are cell nuclei and the nucleus is labeled with this fluorescent protein. And so you can see the nucleus. But what you can also do is you can shine a very bright light on this, and that will bleach part of this fluorescence in the same way that sunlight can bleach colors. And so what they can do is they can bleach half of this cell, and you can see it's gone black, but now what you see is that the fluorescence is coming back again. Okay, you see that? And what that's telling us is that these proteins inside the nucleus, which make up the epigenome, they're actually moving around the nucleus, okay? So because the reason that this fluorescence comes back is because the fluorescent proteins move from over here to here. So the epigenome is plastic and changes over time. But the fact that it changes over time is, also creates a problem for the cell, okay? Because I've told you that, aging comes about because, damage accumulates over time. And so if the epigenome is moving and changing over time, and that means that the epigenome itself is also prone to accumulate this damage. And we can see that happening because, so what we have here is, along this axis here, we've got 665 people who are rank ordered by age from young to old. And then on this axis here, we're looking at the level of one of these chromatin modifications that I mentioned earlier. And all you need to take away from this picture is that this pattern is not random. Okay, if this was random, what you'd see would be a single kind of homogeneous color across this whole box. But what you can see is that there's green here and there's blue here. And so what that means is that these modifications are changing with age, but they're not changing in a random manner. They're changing in essentially the same way in all of these people. And so that means that, and this is work originally done here in San Diego by Trey Eidecker at UCSD. Okay, this is the paper that first reported this. What you can do is you can take this pattern of these modifications and look at it, or more accurately measure it, and from that you can tell how old somebody is. Okay, so from the pattern here we can tell that this person is 75 years old. Okay, or in other words, if we look at that pattern and we calculate from the pattern what we think that person's age is, this person we calculate as being 60, and it turns out that their age is remarkably 60. Okay, so this is what's known as the epigenetic clock. In other words, we can look at the epigenome and essentially figure out how old somebody is. There's kind of an add-on to this, though, which is that you'll see here is that there are some individuals who appear to have what we call an accelerated calculated age. This person, if we look at this pattern, then it says that this person is 72 years old, but in actual fact, they're only 53 years old based upon their chronological age from when they were born. So this person is considered to be a fast ager. And actually, there is some evidence that individuals like this are at increased risk of disease and mortality. And conversely, individuals such as this are relatively slow agers and are at lower risk of disease and mortality. So if you have the choice of being this person or this person, you definitely want to be the slow aging person. The main point I'm trying to make here is that the epigenome does change with age, and we think in my lab that, and my lab's not the only lab that thinks this, that these changes with age are in part responsible for this increased incidence of cancer with age. So these abnormal cells that accumulate here within this tissue, within this aging tissue, these are in part abnormal, we think, because of these changes to the epigenome. And we are, now testing this idea, and the way we're testing it is we're going back to what I told you earlier about this. This is the nucleosome that I showed you earlier. This is this fundamental unit of chromatin. And if we look in there, if we look in a bit closer at that, if we strip some of it away, we can see that there are specific molecular interactions within this nucleosome. And what we can do in the lab is we can actually manipulate these contacts, okay? And we can do this in such a way as to make these nucleosomes either more or less stable. And so that's something that we're doing in the lab. And the question will be is if we affect the nucleosome stability, does that affect the rate of ticking of this epigenetic clock? Can we slow it down? Can we speed it up? Can that affect healthy versus unhealthy aging? Can it affect lifespan? And can it affect the incidence of cancer? And obviously, ultimately, what we would like to do is be able to do this in such a way that decreases the incidence of cancer. And having explored that at kind of a basic molecular level, what we would then like to do is to identify drugs which can mimic this process. And so at Sanford Berman Prebys, we're very lucky to have a really fantastic robotic screening facility, robots which will look, what I mean by screen is to basically take, hundreds of thousands of different molecules or potential drugs and look through those molecules for the molecules which have the specific properties that we're interested in. And in our case, that would be, stabilizing nucleosomes or preventing cancer. And so not only do we have the drugs to do this and the robots, we also have, a fantastic team of collaborators who can help us do this. This is very sophisticated stuff, which my lab is not specifically trained to do, but we can collaborate with Michael, Suzanne, and Thomas to do this. And so the ultimate goal then is to identify a drug which can potentially prevent or delay the onset of cancer. So in a nutshell, then what we would like to do is to take, some of these interventions that I mentioned earlier, exercise and healthy diet, dietary restriction, and, in very simple terms, put them into a pill, which we can maybe take to promote healthy aging and longevity. And so, on paper at least, that's the goal. But I also want to, just kind of, raise a question that kind of goes beyond that, because, there's ethical issues that come into this. Okay, I mean, so do we aspire to be a society which, just sits at home on the couch all day watching TV and eating junk food? In my opinion, that's not a goal that we should be aiming for. So I wouldn't personally advocate that drugs such as this should be, so freely available, so that's that's what we encourage. So there would be questions, if we can identify such as this, the drugs such as this, there would be questions about, who would get these drugs and when would they get them? And I think that's a conversation that society would need to have. And so with that, I just want to thank the people in my lab who do the work. I'm really fortunate to work with a fantastic team of very talented people from all over the world who've come to Sanford Burnham Previs and San Diego, because it is such a fantastic place to do science. San Diego and SBP are known all around the world for the work that they do. So these are the people in the lab. Obviously, we tend to meet, we meet a lot by Zoom these days for obvious reasons. Okay, so this is one of our recent lab meetings. And I also want to thank the people who fund the research, money from the NIH, the Hillblom Foundation, the NIA, Glenn Foundation, and the Padres pedal the cause. And obviously without the money to do this research, we couldn't do anything, because it's obviously not cheap. And with that, I will take questions if there are any. Thank you.
Wonderful. Thank you so much, Peter. I'm going to stop your screen sharing, if you don't mind. This way it's easier for participants to see you. And I see we already have a question. So perfect. So everybody, if you have questions, please feel free to type them into the Q&A function. It's in your control panel. It's got two little speech bubbles. And just type it in there, and I will read them off for Peter to answer. And also, while Peter is going to answer the first question, I'm going to pull up a poll. We always like to survey our audience to see how you enjoy the event. And so just take a moment to fill out that poll, which should be easy to do while you ask questions and listen to Peter's answers. So Peter, the first question is, what are your views on intermittent fasting, especially, specifically 16 to 8 fasting?
Right. Okay. So yeah. So there's a lot of variations on, what's known as intermittent fasting or time restricted feeding. The 16-8 that was just mentioned, I think I would call that time restricted feeding. In other words, all your calories are consumed within an 8-hour period of the day, and that's in contrast to intermittent fasting, which is when you might fast, or not eat, or eat only very little for 1 day a month, or 1 day every couple of months, etc. But in principle, so there's many variations on this, okay? And this has all come about because we obviously appreciate the benefits of dietary restriction, which I showed you, okay? In other words, 20 to 40% calorie restriction is beneficial, but unfortunately, it's not something that most people can do or want to do. Okay, so time restricted feeding, intermittent fasting, the 5-2 diet is another one. Okay, I think where you eat as much as you want for 5 days a week, and 2 days a week you don't eat very much. So I think there are all kind of variations on this theme, and yeah, I think they all make a lot of sense. As I mentioned in the talk, in very general terms, it's thought that the benefits of calorie restriction come about because it really forces the body to put its resources and effort into preservation of tissues and elimination and removal of damage. Okay, and so if we can get the body to do that, whether that's by having a long 16-hour overnight fast, or whether it's by fasting 1 day a month, or 1 day every couple of months, or 2 days a week, I think all of these things can be potentially beneficial. I think what we perhaps need to be careful of is maybe going a little bit too far. And in terms of the time restricted feeding, one of the people who's perhaps thought the most about this is Walter Longo at USC in LA, not so far from here. And I've heard him say that I think he thinks more like 13 to 15 hours is better to go for, that 16 hours might be a bit on the long side, and there might actually be some negative consequences of that. So I think if that's something that you really want to that you're really interested in, and I as I say, I think it makes sense. I would encourage you to, for example, read some of the work from Walter Longo. He's written a book on the subject, and he's a very highly respected scientist who's really translating his work into real benefits. So I would take a closer look at what Walter's got to say.
Thank you so much. And we have another question. What impact does inflammation have on aging? And does exercise and proper eating reduce inflammation, thereby decreasing negative effects of aging?
Yeah, so I mean, inflammation clearly increases with aging. There is something known as inflammaging, which is kind of meant to characterize that general kind of systemic increase in inflammation with age. And I think that the general view is that inflammation is detrimental. There's a little bit of controversy about it because I think it becomes a bit, it's a bit chicken and egg. Okay, I mean, is the increase in inflammation promoting diseases of aging, or are the diseases of aging promoting the inflammation? Okay, so it's a little bit difficult to tease some of that apart, but I would say that overall, on balance, there is there's a general acceptance that chronic inflammation, which accumulates with age in some forms, is detrimental and can promote different diseases of aging. And so, I think many of the interventions that we've talked about, which can promote healthy aging, as part of the process, they do also, decrease this chronic inflammation, and that's probably also contributing to their benefits.
So even for inflammation, exercise and a good
yeah.
diet.
I mean, this is kind of why one of the reasons that aging is such a fascinating and difficult thing to work on and to tease apart is because everything is so interconnected. Yeah. So we can exercise and eat healthily, and that will have many benefits. Okay, it'll benefit many different aspects of our cells and our tissues, but the challenge is to figure out, you know, what's its primary effect. Okay, presumably it's doing some things first and everything kind of follows. But the challenge is to figure out, you know, what's it doing first. Okay, because if we can understand that, then that's where we can perhaps intervene to gain all of the other benefits. Okay, but the reason it becomes, on the one hand, as I say, it's fascinating but also difficult, is because sometimes figuring out, you know, what is the primary effect of exercise is difficult. I mean, I think that's a really fascinating question. Okay, I mean, what is in biology, we like to talk about, you know, what's the mechanism, what's the molecular trigger. Okay, so what is exercise doing at the molecular level, which is, you know, triggering all of these other benefits? Okay, we don't know, but if we knew what that was, then, you know, maybe we could have a drug which could mimic it.
Yeah.
So I can see some of the other questions in the chat. I mean, I can just.
And sorry if I just was slightly.
No, you should go ahead, Andrew, if you want to pick.
Yeah, we're just going to go in order. So the next question was, what is the relationship between damage and telomere length? I'm pretty sure I'm not doing that right.
Yeah, so, you know, again, telomeres are the ends of chromosomes, and they have, you know, many functions in protecting chromosomes from damage and proper function of chromosomes. And what's been noticed about telomeres is that they tend to shorten with age, and there is some evidence that the shortening of those telomeres does contribute to you know, cell dysfunction, tissue aging. And I think it's certainly clear that when telomeres become, you know, if telomeres get very short, or if they become damaged, then they can be a cause of cell dysfunction and tissue dysfunction and aging. Okay, I think that's pretty clear. I think what's less clear, again, is, you know, to what extent, for example, in humans, do we really think that this is a primary cause of aging in humans? Okay, and I think that's where it gets a bit more controversial.
A little bit more complicated and controversial.
Yeah, I mean, there are some specific diseases where it's clear that, you know, telomere shortening is a driver. Okay, I mean, something called idiopathic pulmonary fibrosis, which is a lung disease, which in some respects looks like premature aging. And it's clear that there are some individuals who have, you know, mutations in their telomere maintenance machinery, which can trigger that disease. And so that supports the idea that you know, damage to telomeres can be pro-aging. But it's much more difficult to know whether, you know, in an otherwise, you know, healthy individual, to what extent are the telomeres triggering different types of tissue dysfunction. That's still an open question.
Okay, so we'll have to wait a little bit longer on research to come to an answer on that. And the reason why I was a little bit distracted earlier, and I apologize for that, but I had to look up Volta Longo because somebody was asking for the name in the book. So I Googled that real quick. So I apologize for my quick distractedness.
Okay. I should say, Andrew, I mean, if people have questions, if they email you and you forward them to me, I'm happy to respond by email to specific questions like that.
Oh, great. Perfect. Yeah, and I'll put my email address in the chat again. It's adultprograms@rhfleet.org. It's fairly easy to remember, but I'll add it to the chat just in case. The next question we have, is there an optimal level for exercise for someone 60 and better?
I think it's, you know, everybody's different. You know, because of our previous lifestyle choices, our, you know, as well as our genetics. Okay, I mean, we do age at different rates. Okay, and to some extent, as I say, some of that's down to choices which we made previously, which we can't change, or as I say, our genetics. So I think you just have to do, what you're comfortable with. Okay. I think, different people in their 60s will have different tolerance for exercise. And so, do what you're comfortable with. Okay. I mean, I think if you want to do something, pretty extreme, you want to go interval training on a bike or whatever, I think probably the proper recommendation is to get your heart checked by a doctor before you start doing that kind of stuff. But, apart from that, I would say, just do as much as you feel comfortable with and you enjoy. And that is going to be different for different people.
I think that's a good point. Enjoyment plays a big part, I think, because unless you enjoy it, you're not going to stick with it anyway. So I think whatever makes you happy and gets you moving is probably a good idea.
And if you're not enjoying it, that may also be a sign that maybe you're overdoing it. So I think I think you need to find the type of exercise which you enjoy and do as much of it as you're comfortable with.
Yeah, and that brings up a good question for me. You mentioned that there is the genetics that we bring from our parents, and that's something we can't change. Then we know there is dietary restrictions and exercise that can help, but there's also other influences in the world that will cause aging and diseases and have effect on ourselves. Do you know what the weighting is that can slow down aging between external factors that we don't have control over versus genetics versus exercise and diet?
Yeah, so it's generally thought that about 25 to 30% of lifespan, which is not necessarily quite the same as health span, but about 25 to 30% of lifespan is thought to be genetic. Okay, so that's in our DNA. Inherited from our parents, and the other 3/4 is largely lifestyle based. Okay, so a combination of things like diet, exercise, whether you smoke or not smoke, sleep, all those kinds of things. I think I've been asked this question before about which is more important, diet or exercise. Or something else. I think it's impossible to compare because, there's different levels of all of them. Okay, as we just heard. Okay, a 60-year-old could do a lot of exercise or not much exercise. Okay, so I think, there's and similarly, you can have a very, very good diet, or you can just have an okay diet or a really bad diet. Okay, so it's kind of difficult to say that any one is better than the other or more important than the other. I think the goal should be to just try and, make each one of them as good as you can comfortably.
Great. So maybe not as many Christmas cookies for me this afternoon as I've had in the past week. We have another question for you. What is your view on which, if any, vitamins and supplements are effective with dealing with long-term tissue molecular damage?
Yeah, so as I mentioned, there's a lot of these things being discussed now: metformin, rapamycin, alpha ketoglutarate, nicotinamide, riboside, and some of the other NAD precursors. So all of these are exciting areas because there's more or less evidence for all of them. For example, from mouse studies and some of them even from human studies, that they are beneficial for healthy aging and suppression of disease. In my opinion, it's also a little bit early for all of them. Okay, the reason we have the TAME study that I mentioned earlier is because the TAME study is meant to specifically ask the question: is it better for normal, healthy aged adults to be taking metformin than to not be taking it? Okay, that's the question which the TAME study will ask. Okay, do the benefits of metformin outweigh what might be the side effects? And so until we've until things like the TAME study have been done in a carefully controlled way, it's difficult to really answer though, to really know. I mean, personally, I think that, you know, some of the supplements like, you know, NMN, alpha ketoglutarate, you know, if I was to start taking them, I wouldn't know what dose to take. Okay. I mean, I might be able to figure it out from, you know, some of the stuff that's been done in a mouse, but then mouse metabolism is very different. And so what we know about, you know, biology is that these things are very dose dependent. Okay, you can have a dose which is beneficial, but then a dose which is too low and doesn't do anything, and a dose which is too high and is detrimental. Okay, and personally, I don't feel able to really judge what that proper dose is, and so I personally don't take any of these things because I'm not completely sure that all of the benefits translate to humans without any, you know, without any of the negative effects. And that's why I think things like the TAME study are very important to specifically ask these questions.
So TAME is considered not a drug, but a supplement?
Right, so TAME is an abbreviation of taming aging with metformin.
And metformin is not a drug, but a
oh, so, well, no, so metformin is a drug.
Okay.
Yeah. Yeah, so yeah, there's technical distinctions between drugs and supplements. I think drugs are more, there's probably people who know this better than I do. I think drugs are more tightly regulated by the FDA because they're, you know, perhaps not considered to be naturally occurring compounds as compared to supplements, which, you know, we can probably also get through diet if we want to. So they're less tightly regulated. So there's probably people who may know more about that than I do.
Perfect. Maybe we can find another talk on that. I appreciate you trying to answer that. I really do. And we have a question whether this talk will be available to listen again. And yes, it will be. It will be on the Fleet website, and I believe also on the Sanford Brennan Previs website. But it will take a little while to get it up there. So it will not be available right tomorrow or the day after. I think it's going to take a week or two at least for us to post it. Which is why it's great that you're here live and you can answer your question and you get the information right away. But yeah, we are recording it and it will be available. And with that, thank you so much, Peter, again, for being here with us, for giving this amazing talk and sharing your work with us.
Thanks everyone for listening. Thank you.
And thanks everybody for joining us. And I hope we will see you soon. Thank you so much. Stay safe and stay healthy. Bye.